Today's lecture is on innate immunity. So innate immunity then is what's inborn, and it's our protection against any kind of infection. So it's pretty generalized. It's not really specific immunity. Adaptive immunity is only in vertebrates, matures over time, and basically it takes about 4 to 7 days to fully activate. And the cool thing with adaptive immunity is it does exhibit memory. So the next two chapters, 11 and 12, will deal with immunity, and today we're going to talk about innate barrier defenses and cellular and molecular defenses, and then in the next chapter we'll talk more about adaptive types of responses. So our first lines of defense basically try to prevent pathogens from entering, and these can be categorized as mechanical, chemical, and physical types of barriers. So if we look at mechanical on the left-hand side here, basically you have tears, they wash microbes away. In your lungs, you push things out, they get trapped in mucus, and your skin is a physical barrier. Now chemicals, we do produce things like lysozymes. Also, our stomach is very acidic, and we do have some different types of proteins that will actually destroy pathogens when they get into our body. So, the mechanical barriers, guys, the main one I want you to be studying for your exam is the mucociliary escalator. And what that does is it sweeps away from the lungs and up towards the mouth. Now, we do also have tears, and think of things like urine that goes out, pushes things out of your body. Saliva also limits what microbes can adhere in your mouth. Now, for our chemical barriers, there are several, but please focus on lysozyme. It's found in secretions like tears, breast milk, etc., and it helps break down bacterial cell walls. So it will break down the peptidoglycan that's found in bacterial cell walls. Now, another important one to be aware of is called antimicrobial peptides, or AMPs. Now, these will destroy a wide variety of things like viruses, parasites, and bacteria, as well as fungi. So they can stimulate leukocytes, they can also directly target things like the cell wall, etc., and that's how they will get rid of microbes. Now, a physical barrier, biggest one we've got, guys, our skin. So we have these epithelial cells in our epidermis that are really, really tight. So they resist any type of penetration. So the way you get a skin infection is that the microbe has to penetrate it first. Now, even though our first lines of defenses are good, we still need a little bit more help. And one of the biggest things that we have are our leukocytes, or white blood cells, that can help in that. And that's something that's normally covered in AMP, so I'm not going to go into all the different types of leukocytes here. Now, our thymus is very important, and one of the cool things with that is that it's the site of T cell maturation. Now, our bone marrow is also very important. It's where red and white blood cells are produced. Long bones, like in your arms and legs, are also rich in bone marrow. Now, as far as some of our molecular defenses, one set of chemicals I'd like you to know are called cytokines. They can stimulate things like inflammation, generate fever, actually help to recruit your leukocytes to the site of an infection, etc. So what cytokines are then are signaling molecules that allow cells to communicate, that's huge, with each other, and then they initiate and coordinate immune actions. And there's a lot of different immune actions out there. There's a lot of different cytokines out there as well. Now, another important type of molecule is called a cytophore. Okay? Now, these are organic molecules that pull iron from our iron-binding proteins. So if you think of some of the hemolytic bacteria, those are ones that break down red blood cells, when they break down the red blood cells, they can actually get the iron-rich hemoglobin then inside. So these steratophores help then to bind up that iron. Now, there's another system in our second line of defense called the complement system. There's about 30 different proteins, wow, a lot, that work together in what we call a cascade fashion. Now, what you want to be aware of is that the complement cascade can be broken down into 3 different pathways. So please know the 3 pathways are classical, alternative, and lectin. Those again are the 3 pathways of the complement cascade. So what happens is that all 3 of these get activated in different ways. For instance, in the classical, it gets activated when antibodies bind. Alternative will get activated by directly interacting with the pathogen. So these are important facts to know for your exam. Also, the lectin pathway gets activated when mannose binding lectin binds to the protein. All 3 of these different pathways then activate what's called C3. C3 is very important for you to know. Then that ultimately is going to cause 3 end results: opsonization, cytolysis, and inflammation. Again, once C3 gets activated, you will get opsonization, cytolysis, and inflammation. So what opsonization is then, it tags the invader with complement proteins. And what that does then, guys, is it makes the bacteria more readily cleared by phagocytic cells. So you get these little tags then that get on the microbe, and boom, your immune system recognizes it and gets rid of it. Now for the classical complement cascade, please know it's triggered by antibodies that are bound to an invading agent. The alternative pathway, this one's going to be triggered when the complement proteins activate by directly interacting with the invading agent. So it doesn't really require an antibody. It directly is going to interact with the invading agent. And the last pathway, again, it will become activated or triggered when mannose binding lectin associates with certain sugars on the microbe's surface. So that's important to know. So those are our 3 pathways. That's how they get activated. Now, in our bodies, when we are exposed to something or there's any kind of trauma, a lot of times inflammation occurs. So a lot of different things happen when our tissues are damaged or hurt. It doesn't matter if we scratch ourselves, if you get an infection, whatever, inflammation can occur. So how do you know if you've got inflammation? Well, there's redness, there's pain, there's usually heat, so it's kind of localized where you cut yourself, for instance. You get edema or swelling. And a lot of times you can't really move because there's like a numbness that's associated with the area that's been affected. So basically, we have 3 phases that occur in inflammation: vascular changes, so you get damage there, and basically chemicals will get released during that. Leukocytes are going to be recruited, so we've got to get our leukocytes over here, and then eventually you heal up and life goes on. So please know a couple of the vascular changes. So the 2 main things to know, guys, are that vasodilation is going to occur. So your blood vessels get bigger. Another important thing that happens during inflammation is that you get vessel permeability. And this causes then the blood vessels to be slightly leaky. So the cells themselves, there's little gaps that happen. Then you get some exudate that accumulates in the tissue, and that's like some serum and things like that wind up accumulating. So again, if you take a look here, guys, so you cut yourself, You've got vasodilation, so your blood vessel gets bigger. Then these little gaps form. Then a little bit of exudate or some of the serum comes out. You can also get complement proteins that are coming out as well. Now we have to get some of the phagocytes to where the microbes are actually at. So that's one of the reasons you get that permeability there in the vessels, to allow the phagocytes to get to the microbe. So please know this. Margination is when the leukocytes will slow down and they will adhere to your blood vessel cell wall. Okay, so they're going to start to line up. And then what they do, guys, is they squeeze out of the blood vessel and get to the bacteria, and we call that process diapedesis. So again, guys, margination is when the leukocytes slow and adhere to the vessel, and then diapedesis is when they actually squeeze through to get to the site of the infection. So here's an example. They start to line up, so that's margination, and then when they squeeze through to get to the microbes, that's diapedesis. Okay, and then eventually everything heals up and your skin will eventually go back to normal.